I request Dr. Sumit to check that is it getting aired on YouTube or not?
Okay, ma'am, I'm checking it.
So screen get shared.
Asking me.
Nitesh sir, any screen get shared?
No, sir.
Yes, ma'am.
Okay.
Video
Video is screen. Video is playing.
Sound is clear?
Yeah.
Yes, ma'am. Sir, you are mute. Nitesh, sir.
This is a very nice creation by Dr. Niharika Gokhale, ma'am. Thanks to
Thank you, sir.
you, ma'am. So now I request Dr. Neetu Singh, ma'am, host of today's scientific session. Please proceed further.
Thank you, sir. Thank you, ma'am. A very good morning to one and all, and thank you for joining us. Anyone who stops learning is old, whether at 20 or 80. Anyone who keeps learning stays young. The greatest thing in the life is to keep your mind young. On behalf of Faculty of Pharmacy, Oriental University, I, Dr. Neetu Singh, extend a very sincere, warm welcome to all the dignitaries present here in E-International Faculty Development Program. In this 5-day EFDP, we have high-level professors and colleagues representing many well-known universities and research institutes together with members of relevant international organizations. They have come all across to share their knowledge and enhance our mental horizon, making this FDP a truly international FDP in spirit. The theme of this e-FDP is the integrated approach to the natural resources in drug development and discovery. In the continuation of this scientific session, today we have a very energetic, dynamic personality with us. I am generously overjoyed to take the opportunity to introduce our speaker, Dr. Ashutosh Barve. Currently, Dr. Ashutosh Barve is working as Senior Scientist at Pharmaceutical Product Development USA. PPD is the world's leading contract research organization which works in various key areas of drug development and discovery. Dr. Barve has completed his PhD in pharmaceutical sciences from School of Pharmacy, University of Missouri, Kansas City, USA. During his PhD, he received various scholarships in form of teaching, research assistantship of about $150,000. He is having more than 12 years of experience in various fields of pharmaceutical sciences. He is highly motivated scientist with a strong project management and problem-solving skill. Dr. Barve is a forward-thinking and self-motivated researcher with a strong background in the interdisciplinary area of research such as analytical chemistry, spectroscopic characterization, formulation development, and cell culture techniques. He conducts and oversees research and development projects to create new bioanalytical method, optimize existing method. He is having extensive experience of working in targeted drug delivery strategies for cancer, CNS delivery, novel blood-brain barrier model establishment, and BBB shuttle discovery. He is extensively involved in cutting-edge reactive research from last 8 years and published more than 15 papers in world's top-rated journals such as Journal of Control Disease, ACTA, Biometria, and Journal of Immunotherapy of Cancer. He was awarded with UMKC Dean of Student Honor Recipient in 2018. He had been honored with Multimedia Presentation Award, UMKC Interdisciplinary Conference, Community of Scholar in 2018. Dr. Barve was also awarded with Best Seminar Speaker in 2018, Division of Pharmaceutical Sciences, UMKC. He was bestowed with Outstanding Leadership Award 2014, 2015, 2017 for serving in various UMKC student chapter. There is a lot to talk about Dr. Ashutosh Barve, but due to time constraint and without delaying the session, I would like to call Dr. Nitesh Jain sir, Dean in Charge, Faculty of Pharmacy, Oriental University, to welcome our speaker, Dr. Ashutosh Barve.
Thank you, Dr. Niseen, ma'am. So really, it's a great honor to me, and I'm really feeling very emotional for the welcome of our beloved mentor, guru, friends, and fellow colleague from Bianata College of Pharmacy. So really, with the blessings of Honorable Chancellor, Dr. K.L. Thakral, sir, Honorable Vice Chancellor, sir, Dr. Dhruv Ghai, sir, our Chairperson, e-IFDP, Dr. Garima Ghai, ma'am, Dean Academics, Oriental University, Indore, our beloved Executive Director, Dr. Sri Gaurav Thakral, sir, and definitely on behalf of Faculty of Pharmacy, Oriental University, Indore, it's my privilege to welcome Dr. Ashutosh Barve, sir, as a keynote speaker in International Faculty Development Program. So really, sir, from the bottom of my heart, I welcome you all and thank you so much for accepting our invitation as a keynote speaker in e-IFDP. So welcome, sir. Thank you so much, sir.
Thank you so much, Neetu ma'am and Nitesh sir, for your kind introduction.
Thank you, sir.
Yeah, so may I proceed? May I start it?
Yes, sir.
Okay.
Sir, you can.
So I'll have to share the screen first. How I can bring my PowerPoint now?
Have you opened the PowerPoint before?
Yeah, it is open. It is open.
So, sir, share screen.
Okay. Yep. Okay, now I can see.
That.
Yep. 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 Okay. Are you guys able to see this?
It is getting shared. Yes. Now
Yes, sir.
for
Okay.
presentation mode.
Is it looking okay to you guys?
Yes,
It's
sir.
very nice, sir.
Okay, great. So once again, thank you so much for inviting me. It's indeed a pleasure for me to be here as a keynote speaker in this International Faculty Development Program. And I am really feeling honored to be here. And let's get started with the session. So today's talk will be divided into two parts. Mainly, first part will be the introduction about a technique, and the technique which I will tell you in later slides. I haven't incorporated the name here intentionally. And the second part will be the how we can use that technique and how I used it in my research. So actually, the data which I'm going to show you is not published yet, and we are in process of getting a US patent for it. So I may not give you all the details, but I will try to show you that how you, if someone want to use that technique for their own research, they can get an idea easily with the amount of data I will share with you guys during this presentation. So let's get started. As you have chosen a very good theme for this uh, faculty development program, and that is the natural resources in drug development and discovery. So whenever I actually, before I worked on that particular technique, I used to always think about the natural resources for drug development discovery, mainly is from the plant sources. We always had that pharmacognosy subject, which we studied a lot during our bachelor's and master's degree. And we always had an impression that natural means something related to plants, mostly in most of the cases, although there are some animal sources and also some mineral and microorganisms, as penicillin we got from the microorganisms. So we had an idea about those all stuffs considered as a natural resources for the drug development and discovery. But today we are going to learn, I'm going to discuss with you guys about a comparatively less common, a little bit newer compared to the other established sources, but very powerful resource for new drug development and discovery. And I hope you guys will enjoy this session, and let's keep it casual, no need of formalities. So if you have any question, any doubt during the presentation as also, you can just ask me on that same time. So I hope that Dean Pharmacy, Dr. Jain, and Dr. Niharika Gokhale will not mind if I will have a small quiz in starting rather than starting with some big technical
No, sir.
things.
It is absolutely okay from our side, sir.
Thank you so much.
No,
So as you
sir,
can,
if you are coming with something, no, so that will be innovative or innovation will be there. So I know it previously.
oh, really? I didn't know that. Okay, thank you for your compliment. So let's see. This is, I think everybody knows our great nation, India. So let's have some quiz. It will be starting with some easy questions, then we'll go for a little bit tough questions, and I will appreciate your interaction during this quiz. I will appreciate if people will uh, come up and answer the questions. So my first question is, what is the India's population? Anyone knows? Any idea? Any guess? Or I think you must know it. I think we are on the top of the list, almost top of the list for the populations. Anyone knows what is the India's population? No one knows?
Exact figure we don't know, sir.
No, it's no need of exact, just about, I mean approximately.
Approximate 135
About
crore,
6.
130 crore.
Exactly, 130 crore, yeah. So it will be about 1.35 billion people, right?
Yes.
Yeah, so this was a easy question. Let's move a little harder one. Okay, so this is our planet Earth, right? And if I will ask you that what is there is some problem in animations, it seems. Okay, are you guys able to see the question on the top of the screen?
Human population on Earth. Just asking.
Yeah, but I cannot see it because of something
And the
is there
answer is
on.
also visible, sir.
Yeah, now I can figure it out. Okay, so yeah, let's go back. Forgot about the answer. Let me ask you a question once again. Okay, so what is the human population on Earth? Don't say that answer, okay?
Sir, it is 7.5, sir. I'm not.
Yeah, it is about 7.8 billions. You are very right. Mhm. We can write that 7.8 billion also in this way, and I'll tell you shortly that why I'm writing it like this. I just want to get rid of this tools screen. I cannot remove it from the screen, I guess. Okay, that's okay. Okay, so yeah, so the human population on Earth is about 7.8 billions. Let's go to the next question. Now, my question is, what is the most common entity or creature on Earth? I mean, as we know that humans are 7.8 billion, but we are not the one who are ruling in terms of the numbers. There is some species, many species as such, who are more than human beings in numbers. Do anybody know that which entity or which creature on Earth is the largest in numbers? And when I'm saying on Earth, that means any part of the Earth biosphere. It is atmosphere or hydrosphere or lithosphere or anywhere. Any guesses?
Plants.
Plants, no.
Microorganism.
You're partially right. Who is answering it? Is it possible I can see the faces of whoever is
Dr.
attending this?
Dr. Sumit Dwivedi sir is answering.
Okay, sir. You are partially right. Before when I was thinking about it, when I didn't know about this fact, which I'm going to tell you in a couple of minutes, I thought about ants. Did anybody else also thought about ants?
Yes.
Yeah, so they are also one of the, big contenders, and if you can see, their numbers are like 10 raised power 15 as compared to 10 raised power 9 for human beings. That's why I kept it in like this. So to keep the things in perspective. So if you can compare, they are much larger in numbers compared to humans, but they are still not this is not the right answer actually.
Okay.
The answer is not yet revealed. So any more guesses? If not, let me tell you, it is phages, or they are also called bacteriophages.
Yes.
They are ruling our planet in terms of number, and we are nothing in front of them. Yeah, so this is the population of phages. You can see it's 10 raised power 31 compared to 10 raised power 9 or 10 raised power 15 for ants and humans. So we cannot even imagine that how big this number is. To again keep the things in a perspective, I can show you that if we will count all the organisms on Earth, then this blue part will be the number of phages, and this pink part is rest of all the species together, combined together. So you can see that how big their population is in terms of numbers compared to all other organisms. I hope this must be, you guys will find it interesting. And actually I found it interesting, so I wanted to incorporate it in my presentation. And our today's topic is also related to phages, so this was making sense for me to put it in here. And if you will ask me that where someone can find and where they live these phages, so the answer is they are everywhere. They are in oceans, they are in mountains, they are on insects, they are on plants, they are on humans. And still, when we are talking, billions of phages are on our body, including in our eyes. They are everywhere. Wherever you can imagine, they are there. And in unimaginable numbers. in billions of them are on your hands and on your eyes, in your intestine, everywhere. So then question arises, what is phages? So phages are actually viruses. Don't worry, they are not like COVID. They can only infect bacteria. These are the viruses which can only infect bacteria, and they are not harmful for human beings. How they looks like? They are very simple living organisms, half living, half non-living, you can say, because they don't have hands, or I mean, I was just trying to make a let's forget about it. So they are not living, but not dead as well. So they have just two things. One is genome. So genome is either a DNA or RNA, single strand or double stranded, and that genome is covered with a protein coat. Just two things, and this whole thing is called as phage. And they may have some genes right from number 4 to hundreds of genes in their genome. Okay, so we'll not go too deep into that. One interesting thing which I wanted to share with you guys is the name phage. So whenever I work on something new, I always try to find out that why that particular thing is called by that particular name. So why they are called phage? Why not something else? So when I was researching about it, I came to know that this phage word was derived from a Greek word called phagein, and which means to devour, or in simple terms, eat hungrily or quickly or destroy completely. So phages are something which eat up something or destroy completely. And when we are talking about bacteriophages, so bacteriophages will eat and destroy bacteria. So they are the enemies for the bacteria, not for humans. So we don't need to worry about them. Even in some way, they are helpful for us. So how they looks like? They comes in many different shapes and sizes, mostly in the nanometer range. And so this is the pictorial presentation, and these are the actual pictures of those phages under electron microscope. And for today's lecture, we will be concentrating on this phage M13 filamentous M13 phage. How we are going to I'm going to tell you in later slides. So let's go ahead. Okay, now you guys might be agree with me that okay, phages are cool, but how they can help in drug discovery and development? Yeah, that's a big question, and that's the main question here. So the answer to this question was given by Dr. George P. Smith in 1985. What he did is he first introduced the phage display technology. And because of his work on this phage display technology, he got a Nobel Prize in Chemistry in 2018. And I'm very fortunate enough that I was, and even it's a matter of pride for me that he is professor in my university, and I got a chance to see him after he got this Nobel Prize. And I completed my PhD from the same university from where right now he is a professor, and he did that work which worked as a Nobel Prize winning work from the same university, that is University of Missouri. So what he did is he actually engineered or modified the phage code protein using simple recombinant DNA technique. So if you remember the previous slide, I showed you that phages are consist of two things, genome and the protein code. So what he did is he modified this protein code and did something to it. What he did, I will tell you in the next slide. So the technique, which I mean, I didn't mention it in the starting that what technique we are going to discuss today. That technique is the phage display technique, and this technique uses phages, which you know now what is phages, with some unique display features. So what is this display is? I'm telling you in the next slide, or even in the same slide. So as I mentioned that Dr. George Smith did some work on protein coat of this phage, and because of that modification, some sort of things display on the surface of the phages, and that's why this technique is called phage display technique. So how he did that, I can show you. So what he did is he took a phage and with the help of some biotechnological methods, he inserted some diverse set of genes in the phage genome. And because of this insertion of gene, we can say the gene of interest for us, when those genes were inserted in the phage genome, some character came up or some characteristics have developed on the surface of the phage. You can see here. So previously phage was like this with his own genome. And when we inserted some genes, so because of this insertion of this blue part, some sort of character came up on the surface. And this character gives us an example of genotype as well as phenotype character in that phages. And by changing that set of gene which we incorporate here, if we will change them, because we can decide that what we want to insert in the genome of phage. So by changing that genome, the set of gene, we can get different types of characters on the surface. So by combining all different types of sets of characters on the surface, we can make a library of phages with some hundreds of billions of different characters on their surface. So this thing is called as phage display library creation. So this is how he made a phage display library. Not him, but his ancestor, I mean, in terms of his research. So now this phage display library is available commercially. So what are the types of phage display library available these days? So on basis of the gene inserted in the genome, we can get different types of phage display libraries. So if we are inserting gene which can make or create a peptide sequence on the surface of a phage, then we will call it as a peptide phage library. Similarly, we can insert genes which can produce antibodies on the surface, then it will be called as antibody phage library. Similarly, for SCFVs, we can get SCFV phage library, so on. So there are various types of libraries available, but today we are going to talk about the peptide phage library. So for peptide phage library, still again, I mean, there are different types of peptide phage libraries also available. So if I'm saying PhD7, that is PH means phage display, phage, D means display, and 7 means number of amino acids. So peptides are kind of aggregation. If we will join too many amino acids, that will be called as peptides. So if there will be 7 amino acids, then we will call it as PhD7, I mean peptide phage library 7. Similarly, if amino acids are 12, then it will be called as PhD12. Like C7C, if some cyclic peptides are available on the surface, then we will call it as PhD C7C, and many more are available. But this is just to give you a idea that how many different types of peptide phage libraries are available. And this is how it looks like. Sorry. Okay. So this is commercially available, BioLab New England. So this is how it looks like. The liquid you can see, this is about 100 microliters, contains about 1 into 10 raised power 13 PFUs per ml. So PFUs are number of virus particles or number of different phages. So you can see here in this library, you will have about, if you have a 100 microliter total volume, then you will have about 10 raised power 12 different phages with their different display characters. So you can see how big number is. If you remember, the human population, it was about 7.8 10 raised power. How much? Is everybody awake?
Yes.
I will try my best to keep all you guys awake, but it's your responsibility as well to be awake. Okay, let me check what was the number.
9.
So 9, sir.
Yes. If you have told me this earlier, I didn't have to go back. And it's for 9, right? So as in this 100 microliter of uh, okay. In this small vial, we will have phages more than human beings on Earth. Okay, so how powerful it is, you will realize it in later slides when I will show you that how you can use it. Let's go ahead. Now you will say, okay, phage libraries are exciting, but still you are not sure that how someone can use them in the drug development and discovery, right? So let's see how. So what happens is, if you are aware about any type of research, then most of the times, if we are treating some disease, or if we are, so if we are treating any kind of disease, we generally encounter some sort of either a protein or a receptor or a different type of cells. Most of the times, these are the targets of most of the drugs which we use generally. So if we want to find, if anybody of you have chemistry background, then you might be aware about the combinatorial chemistry, in which we use several different hundreds of different molecules against one set of target. So similarly here, we can use our peptide library. So in case of combinatorial chemistry, you can maximum you can go for like thousands of different lead molecules, but here you can have hundreds of billions of options. I mean, hundreds of billions of different sequences which you can try against a specific target. So peptides are also kind of drugs. So if you know that what is your target, then you can easily do a experiment in which you can have that protein or receptor or cell, and you can you can throw all of your peptide library on that to check that which particular uh, peptide sequence on their surface can be able to interact with our target of interest. So if you can see, similar to the receptor, that lock key concept, similarly here also the peptides can interact with the target, and we can find those particular sequences which have the capacity to, or which have the affinity to interact with the particular type of target. So what we can do is we will have a target with us, and we can put our library on it, and we can check that which one will allow them to interact, and then we will do the washing. So only those which have some sort of affinity towards the target, they can bind to that target. Others can be easily washed out. In the next step, what you can do is you can elute out the leads which you get from this experiment, and those leads you can make, I mean, you can amplify the numbers of those particular phages. Why we need to amplify them? Because we cannot just work with one phage. If we want to, I mean, if you want to know that what is the particular sequence here, peptide sequence, which has the ability or which has the affinity with our target of interest, then you need to, I mean, do there is a thing called uh, sequencing of peptides, so sequencing of DNA. So what we generally do is we collect these phages, and then we remove the DNA from the phages, and we can check that what is the sequence of that DNA. On basis of that sequence of DNA, we can check that what is the particular peptide sequence there which has that affinity for that target. So in this way, we can find the correct peptide sequence, and we can use it in many ways. Like targeted drug delivery is all about finding target and binding it with the target. So peptides and proteins are, as you may know, that is the hot topics from last couple of years. So this is how you can use, I mean, one library gives you a chance to check billions of different sequences in very short span of time. So I hope that you guys are understanding that how powerful this method is. Okay, let's see next step. So this was a kind of introduction that how this method works and what is the principle behind that method. And now I'm going to share with you guys a little bit of my research, which I did during my PhD. It was one of my project on blood-brain barrier shuttle peptide. So it was a discovery of blood-brain barrier shuttle peptide. So let's start with a little bit of background about the blood-brain barrier. So actually, blood-brain barrier is a physical, chemical, and metabolic barrier which restrict and control the movement of substance between the general circulation and the brain's extracellular fluid. It makes a kind of it's a kind of a barrier which makes the brain as a restricted area, and only authorized molecules can pass through that barrier and can enter into the brain. The blood-brain barrier concept first time was, the work which helped us to understand the blood-brain barrier. The work was initially done by a German scientist called Paul Ehrlich in 1885. So what he did is, actually he was doing some staining experiments, and what he did is he injected some dye in a rat and through IV. And what he found is that all the organs in the brain's body got stained except the brain. Although at that time he was not very sure that why it was happening, and because he didn't have any understanding of blood-brain barrier as such. So, but he pointed out at that time that the brain didn't get stained properly compared to the other organs of the body after giving the IV injection of some staining solution. So here the blue color represents the stained region, and the gray color is the unstained region. Later on, one of his students, his name was Edwin Goldman, and he did another interesting experiment. What he did is he directly injected the dye into the brain of that rat. Now what he observed was that only brain was got stained, but not any other part of the body. So with this set of experiment by him and by his professor, they came to conclusion that there is some sort of compartmentalization between the brain and the rest of the body. But still they were not very sure that why it was happening, because they didn't have any technology at that time to see the difference between the brain vessels and the other parts of the body, the vessels present in the other parts of the body. So, but later on, with the better technology, as you can see here, this was one of the paper, and I found this picture very interesting. If you can see here, it is an autoradiogram. An adult mouse was sacrificed after the injection of radio-labeled histamine. As you can see, so it was intravenous. So again, you see the black part is the presence of that radio-labeled histamine, but the white part shows that there is no histamine present there. So if you can see that here, it is clearly shown that brain and the spinal cord doesn't have any sort of radio-labeled histamine present. Although the other parts of the body, all you can easily, you can see it easily that they are there. So here again, this was a kind of example that how the things doesn't go in the brain very easily compared to the other parts of the body. So let's see this video. I hope everyone is awake.
Yes, yes, sir.
Yes, sir, definitely.
Good.
Yes, sir.
Is there any sound, sir? Because
Yes, you can't hear it.
no.
Yes, sound we are not able to hear, sir.
Share
I
your
don't know.
share your computer sound.
Let me try how to do that.
When you shared your screen, no, at that time only computer sound should get shared.
Let me try. Oh, okay, got it. Yeah. Is it working now?
Yes.
Yes, yes.
Yes.
Okay, great.
The blood-brain barrier, such as other blood barriers, is a remarkable anatomical adaptation. Its most striking structural feature consists in endothelial cells, tightly joined by tight and gap junctions. These structures bind the cells side by side through several proteins, including connexins, claudin 5, and ZO1, thus maintaining the barrier's integrity. Pericytes cover about a third of the capillary surface, and surrounding glial cells, named astrocytes, support these capillaries with their foot processes. This capillary bed in the human brain has a total length of approximately 600 kilometers, with a surface area of about 20 square meters. It is permeable only to some molecules, such as nutrients entering the brain, or waste products, hormones, and excess neurotransmitters that leave the brain. Only a small number of drugs are able to translocate it. To be able to do so, these molecules require lipophilicity, a size smaller than 500 Daltons, and evasion of mechanisms of active extrusion.
All right. So what that video was saying, we can summarize here. So the blood vessels present in our brain are not same as the vessels present in the rest of the body. I mean, the arrangement of cells in the vessels is very different compared to the other parts of the body. How it is different, I can show you here in this picture. So if you can see here, this is the type of cell arrangement in the blood vessels in the other parts of the body. If you can see here, there are some clefts and fenestrations and gaps are available, and only one layer of cell, that is called endothelial cell, is present. However, in contrast to that, in brain, if you can see, there are several layers of three main different type of cells, which makes a kind of tight junctions, and there is no gaps between them, no fenestrations available. So that is why the things cannot penetrate out of the blood vessels into the extra cellular, I mean the inside the brain area. Only it can be in this lumen area. However, in the other parts of the body, that's why the drugs and whatever, I mean, if we are taking some medicines, they can easily penetrate through these gaps, but they cannot penetrate through here. And this makes the blood-brain barrier very difficult for anything to get passed through it. Okay. So this is our brain. So what we learned from this is our brain, and when something harmful or not good for our brain tries to enter into the brain, then blood-brain barrier comes into the picture as a savior, as a hero, and keeps our brain healthy and happy, right? So that's good, right? What is the problem then? The problem arises when our brain gets some disease or some infection and we want to treat them with the help of some medicines. Here also the blood-brain barrier comes into the picture, and it will not allow any drugs to treat our brain. And this is what is the problem. And this makes the drug delivery to the CNS as a nightmare. I mean, almost 100% of the large molecule drugs and almost more than 98% of the small molecular drugs cannot cross blood-brain barrier. And this is the reason why a small infection in brain can be life-threatening. Although we have a lot of other antibiotics or drugs available which we can use to treat or to kill those infections, but we cannot let the drugs cannot penetrate into the brain, although the infective organisms can somehow they can manage to go into the brain by some certain unknown pathways. So this makes these CNS disorders such as Alzheimer's, Parkinson's, brain cancers, even in case of cancers, the stem cancer cells, they migrate into the brain, and if you do the chemotherapy, that stem cells migrate into the brain and you cannot reach in the brain. So once your chemotherapy will be over, that cancer cell will come back into the mainstream and again start making new cancer cells. So this is one of the very difficult to treat those type of cancers. And this is how the CNS disorders largely unmanageable or undertreated. So it's not like nobody has worked on in this area. There are several different types of techniques have been used. Many times, what is the idea behind their CNS drug delivery is to have invasive techniques. Invasive means they disrupt the blood-brain barrier. They disrupt the cellular arrangement in the vessels in the brain, and then this is how they wanted the drugs go into the brain. But at the same time, they are making our brain open for the harmful things. So that was not a very good technique to do. Many non-invasive techniques have also been used, and many big pharma companies are spending billions of dollars to get something good. I mean, which can easily, without having any side effects or without having any issues, can deliver the drug into the brain. And there are several techniques have been used through intranasal or many others, but still there is no gold standard have yet been set. There is one area which was not, studied very deeply, and that was using peptide as a carrier. So peptide, as now you can reconnect that why I'm saying you all about those peptide phage libraries, because I have used those peptides to check if some can go through the blood-brain barrier and work as a carrier for the drug delivery to the brain. So here we chose to go for the peptide as a drug delivery carrier for the delivery to the CNS. So just to refresh your memories, so this is amino acids. I think everyone must be aware about what amino acids are. And when they add together, they join together, then they can form peptide. And up to certain length, they will called as peptides. But if they increase in numbers, then they will called as protein. So we decided to identify some peptide sequences which can work as a carrier for drug delivery to brain or CNS. So we used here the 7-mer peptide phage library. As I said earlier, that we have billions of different peptide sequences available, but I mean the crucial question was that how to screen them. So we designed a very novel screening system for screening phages from the peptide library to get a perfect candidate. So what was the design? I'll not go in details, but it was consist of both in vitro and
sorry
in vivo.
to disturb you, Dr. Ashutosh Barve sir.
Sure.
Sir, actually, we have another session from 12 o'clock. So if it is possible, so can you conclude this session within 5 to 10 minutes, sir?
Yeah, sure. Why not? I can. Was it too much of time I took? Sorry.
No, sir.
Okay. All right. It's not much left. So it was consist of actually 5 rounds of selection. Initially, 2 rounds were with the in vitro method, and rest of the 3 were about in vivo phage selection process. And we prepared some transwell assembly which can mimic the actual blood-brain barrier system. And we used our peptide libraries to, we use the peptide library to get through those models, and it confirmed the workability of our system. We did some tests. It's not that important, but what we did several rounds of phase selection process. We did in vivo as well, and so this is how the 3 rounds of in vivo took place. And if you can see here that after the first round, only 5% of the phages which we incorporated in the first round can pass through that our in vitro model, which confirms that okay, there is some sort of selectivity in the phages. Not everything is going down, but after amplification in the second round, it was increased. As I said, amplification took place, so it was increased. Similar thing happened in the in vivo. So as I mentioned in the earlier slide that we did the DNA extraction purification and then sequencing to get the peptide sequences, and we got about 7 highly repetitive sequences which had a potential to pass through the blood-brain barrier, and this was the most important slide. So what we did is we made it 2 groups. One was with rhodamine alone. I mean, rhodamine is a dye which is known that which cannot cross the blood-brain barrier. So we used one group of animals with dye only, and another one we conjugated that dye with our peptide which we identified, and then we did IV injection and sacrificed the animal and collected the organs. And if you can see here red colors means the presence of the dye. You can see there is no red color in the brain in case of first group, but in second group you can clearly see the presence of red color, which shows that because of the presence of this peptide sequence, that rhodamine easily gone into the brain. And recently we were able to make a formulation also, which was able to, go into the brain with the help of our peptide sequence. And this was my lab back in PhD. And I would like to thank to my professor, Dr. Kun Chang, all my committee members, my lab mates, friends. And most important is these guys, because they gave their life for my project and for many of the projects for human beings. So thank you so much for your patient listening. And I'm sorry if I took longer than what I was expecting.
No, sir, you have finished on time actually.
I'm done.
Yeah. Thank you so much, sir. So thank you, Dr. Asutosh Barvi, sir, for your really very beautiful presentation. You have presented very good thoughts of research in a very scientific manner. As always, we admire your research, your knowledge too much in our life. So really, this presentation was too good for all the research scholar and the participants. So now I request.
You are too kind, and thank you so much for your invitation, and it's all my pleasure to be here.
So Dr. Neetu Singh, ma'am, yeah, please. नीतू सिंह मैम आप अनम्यूट कीजिए. Yes, ma'am, please proceed.
Thank you, sir, for such a valuable and enlightening presentation on novel topic, phase display technique. In such a graspable manner, it was really pleasure to hear your session. Now, I would like to request Dr. Nitesh Jain, sir, Dean in Charge, Faculty of Pharmacy, to facilitate Dr. Ashutosh Barve, sir.
So again, it's honor, second honor of the day that I get a chance to appreciate, and it's a token of love which I'm presenting to our honorable beloved mentor, Dr. Asutosh Barve, sir. So, sir, on behalf of Faculty of Pharmacy, Oriental University, Indore, I have presented this certificate as a token of our love, our support, as accepting our invitation as a keynote speaker in International Faculty Development Program. And really, you have done a great job, and you have given a lot of information about the basics research. And we always feel proud to that our colleague, our mentor in USA and representing India. So Dr. Ashutosh Barve, sir, thank you so much, sir, for your kind.
Thank you so much, Dr. Nitesh Kumar Jain, sir, for your kind words. And I would like to congratulate all of your team for this great event. And I know there will be a lot of hard work and efforts which came together to make this
sir, all credit goes
successful.
to my dynamic team, sir. Definitely.
Yeah, I know that. Yeah, very good. I mean, well done.
My team, but actually it's all my teamwork. They all are dedicated, committed towards the development of Faculty of Pharmacy, sir. So whatever whatever you have given in 2012, so just I'm maintaining the same decorum, and we are trying to upliftment of pharmacy department in a very good way. So we need your blessings forever, sir.
No, I know you are too kind. I mean, every time, whenever you say something, your kindness comes out of your words. So thank you so much for your kindness and everything. Yep.
So thank you so much, sir, once again. So I requesting Dr. Niharika Gokhale ma'am, please conclude this link. And if you want to say something, so you can say, and we can move further for another session.
No, sir, it's better to move further for another session. If
Sure.
Sure, sure.
something,
So
no,
thank
I
you so
will
much, sir.
Sure,
talk
sure.
to him personally.
Sure.
Ashutosh.
Sure.
Thank you, sir.
Okay, thank you, sir.